Recently, we had the African BioGenome Project event. Could you tell us more about this event as well as Separation's involvement?
Okay, thank you very much. Yeah, we had this exciting event that is ongoing. As we are speaking, we already have participants that are at our Separation demo lab, whereby we had actually to do hands-on experience for them, whereby they had to sequence some genomes that they prepared from the start to the end. And this is very exciting because we have seen a lot of genomics works going on around Africa, but unfortunately, the only African thing about them was the sample that were collected from Africa. But now with the Africa BP, we are trying to make sure that every genome is sequenced locally, actually by the local scientists. So this is very exciting. And what I'm more amazed about is, after taking the participants through the whole workflow, you can see the excitement, and they are all eager to go back to their different countries and actually put to action all the lessons they have learned from this experience.
So Africa BioGenome Project is an initiative to sequence more than 100,000 genomes of different endemic, indigenous and agricultural important plant and animal species on African continent. The idea is that we build and enable building capacity of genomics in Africa.
Why are you so passionate about the development of African genomics?
I'm passionate about this because it's no doubt that Africa has a very rich and unique biodiversity, and it's very important for us to understand that biodiversity in order for us to conserve it. And also what excites me is that it's going to be developed by Africans in Africa. So that's very exciting for me right now.
All right, it is known that about 95% of the known endangered species in Africa haven't been fully sequenced. And again, there's a problem where over 70% of the studies that actually focus on improving the biodiversity in the continent has been led by scientists from other countries. So this African biogenomics is just here to circumvent that problem and at the same time actually build the much needed expertise in the continent.
For me, the passion comes mostly about who's doing it, because it means we're finally owning the narrative. We finally correlating the data, and we're living in a data revolution. So we are in control of what data we need to generate and how we're going to use that data to improve Africa.
Well, what's there to not be passionate about? Africa has a huge range of biodiversity, from land animals to marine animals surrounding the coastline. And while a lot of people in the world know the select few animals that Africa is known for, there's so many that are not known, and so many that are endemic to Africa that there's no genomic information about. And that leads to a possible loss of biodiversity when natural human processes, industrial processes, urbanization, or even over-harvesting of creatures and over-cultivation can lead to a loss of biodiversity through even extinction. And one main thing in conservation of biodiversity is genomics, and that's the starting point of conservation. You have to know.
Okay, there's a phrase that says charity begins at home. So my biggest objective is to put Africa as a continent on a map. And secondly, when we do policy making, we based our information on the researches that have been done in Western African countries. But considering that Africa is an a diverse country and so unparalleled, I think doing researches based on our biodiversity will help in making best decisions for policies, and then also increase yield in agriculture. And then thirdly, we will have our own information that we can use in academia. Thank you.
Could you please tell us more about why Illumina is such an active role in focusing on developing African genomics?
Illumina understands the need to build the capacity for genomics in Africa, because they understand, one, the biodiversity in Africa, and how it can better advance our knowledge of the human genome to better improve human health.
What key insights have you gained from your genomics workshop and experience with Separations and Illumina?
I've gained quite a lot. I've broadened my understanding of genomics and also enhanced my technical skills in sequencing by being exposed to the latest sequencing platforms. And this makes sequencing less intimidating for me.
Okay, what I've gained is that there's still more lot that I need to learn. And what I can say about Illumina and Separations are that they've got burst technologies platforms that are offer high performance, and you get your results at reduced time and cost. And the other thing that I've learned is that their technologies can offer a vast range of needs depending on the researcher's interest and sense of urgency. And those platforms, they comes with a variety of different protocols that you can choose from.
Okay. Over the last 3 days, we've really had some intensive hands-on experience on how to actually manipulate or handle the DNA sample down to the library construction, the quantification. And the thing that really stuck out the most with me was the data analysis part of it, which was very, very impressive. So I really, really enjoyed this past techniques.
Well, generally, when I've had to sequence samples, I've had to send them off to be sequenced. So this was the first time that I've actually been able to be hands-on and seeing the process of sequencing. So it was really quite amazing to see the translation of theory that you learn into practice.
Most insightful thing that I've realized in this past 3 days is how to make decisions. As a mid-career scientist, we often make decisions that are about money. How do I buy the next piece of equipment? How much money do I have? And being in this practical workshop, I've realized that there is so much more that needs to go in there. And I'm so impressed because Separations took the time to actually give us all this information on how to actually make an informed decision. I don't just buy a MySec because that's how much money I have. I have to think about, what do I want to do? Where's my research going? If I'm going to use this machine for the next 5 or 10 years, then what machine do I need? If I don't have the money right now, which I don't, I have to wait for another year, raise another grant, and then buy the right piece of equipment. Whereas before I came to this, I was so quick to just sell me what I can afford. And I think for me, that is invaluable. As I build my research team and my career forward, I think that was so eye-opening for me, and I'm so impressed.
How has this workshop benefited African scientists?
I think they really enjoyed hands-on experience. We exposed them to end-to-end Illumina solutions. We did library prep, sequencing, and data analysis. And I think for some of them, they even sequenced their first genome, which I think was very exciting.
From the feedback that we received from the delegates, it seems that they've really enjoyed the workshop. Most of them said that it's amazing to receive this hands-on training. It also enables them to do library prep in their own lab. People tend to feel that library prep is too difficult to do, and hence they send their samples overseas. So this also helps them to prep their own samples and keep the samples within Africa.
I believe Illumina is running a Greater Good initiative. Could you tell us more about what that entails?
Sure. The Greater Good initiative is an initiative that was started by Illumina a couple of years ago. It's a fantastic initiative that grants individuals up to $350,000 to better advance their genomics projects, with a focus on agri-genomics, but it's a great fit for the African BP project, because it gives a foot in the door for young researchers looking at advancing genomics in Africa and advancing their projects.
Do you envision that there will be more workshops hosted by Illumina and Separations in South Africa?
Absolutely. After seeing what the outcome for this has been, definitely it's an initiative that Separation as a company is taking very seriously, and we're going to have even many more workshops. For this one, we actually focus on the larger genomes, but we're planning even to do other work involving like microbial work. And as you're aware, I'm one of the many application specialists we have, and I travel quite a lot, and seeing what we are able to do here within a short period of time, definitely we want to bring more scientists here so that they can learn as many different techniques from the experts. So definitely looking forward to that.
Now, if someone's interested in getting this hands-on training, how do they go about it?
So we've got a training academy here at Separations, and we're very passionate about hands-on training. So you can either come directly to our lab or we can come to your lab. Please send an email to training@separations.co.za.